tip that into the funnel very carefully. So you hold the funnel and tip it in slowly and carefully. And what should happen is that you should get a fluorescent layer coming up underneath the saline layer.
Oh, have we filled the funnel with water?
Yeah.
Right, because we don't want any air.
Huge.
Pour carefully, try avoiding any air bubbles.
Nearly impossible.
So you need to get the funnel quite full.
Not so tight that it restricts breathing.
Yeah.
Oh, she's much better than I am. It will settle down because it's denser, more dense. We'll make a Manhattan cocktail spritz.
You can adjust the belt until you're happy with what you can see. You'll be working in pairs. One of you will be the guinea pig and one of you will be the experimenter. Once you're happy with the fit of the belt,
Slowly remove.
Oh, that's worked quite well.
That's worked quite well.
Yeah.
Now,
Can you
how
move
we
it
want...
down and see that there's two separate layers?
Yes, can you all come and see that there's two separate layers?
This is because either consciously or unconsciously making a fact what is recorded.
Okay.
Okay, we're ready for the red layer now. It's got red food dye in it. If you do the same thing, pour it slowly into the funnel.
So try avoid any air bubbles. So pull quite
Recording
steadily. Keep going.
onto the power, onto the laptop. It's quite a useful piece of software, but there are some controls that you will need to be familiar with. First one is start stop.
I went a bit easier on the food.
Yeah,
This as you might expect, starts and stops the recording. You might want to adjust the vertical scale so that you can get a whole trace onto the screen. If you go over to here and adjust the vertical scale, in this drop down menu,
Is it looking like a Manhattan
there's an
cocktail?
option there called auto scale.
Red on the bottom,
Quite well. If you go down
green in the middle, and clear on top. Or is it called traffic
You can
lights?
adjust the time axis.
I don't know. I invented a cocktail a bit like this once. I don't know what was in it, but...
Collins bar, which only works when recording is being made.
So the principle would work with any fluid. So when you're making a cocktail, that's what you're doing. When you're making a cocktail, it's not salting. Normally it's more sugary fluid, it's more dense and will go to the bottom. So in principle, any fluids of different densities will
Wow,
sound
that's
layers
impressive.
like that. As long as you pour them quite carefully and pour the heavier one in under the light
Now
one.
they're quite jumpy. See if you can catch a couple. Okay.
But then drop it in from above. Don't let your net fall.
Take the funnel out now.
Take the funnel out.
They're in this
You
one.
want to have a look at
You might have to take the lid off.
the data you've collected. You block out
So the colours are just so you can visualise the different densities. Obviously a rock pool doesn't normally have these colours in. So, but certainly that sort of stratification of salinity you find in a rock pool, right?
Pull, yank it off.
It will tell you
Do you want to have
These rid of.
It means that you can have a look at how
There
deep
we go.
it's not directly.
How many do you want?
Just one will do.
The instruments on the
Me
colour.
too, if you can catch them.
But it will tell you whether you're breathing deeply,
We tried with our hands, but they've got these very sharp.
Yeah, they don't like being picked up.
your breathing rate. There's also
Great.
one called suction duration,
Okay.
which is the time
That's in
Yeah.
charge now.
See in there? And there he goes.
Right, let's watch.
Okay.
You can try and video him, video his behaviour.
Usually displayed.
And we need another piece of apparatus going, don't we?
Yes, we need to measure the salinity and find out what salinity actually
We're going to
likes.
save the data that we collected. Again, this is explained in the schedule. So follow those steps to make sure the data doesn't get lost.
Who's going to monitor the salinity? You've
Now
not
talk
heard
about
anything
what
yet.
you're actually going to do during the practical.
Put the probe gently in one corner.
No.
Anytime you're doing a physiological experiment, you want to
Tell us what the salinity says when it's stabilised. Oh look, he's already going into
Yeah,
the...
I think this is working quite well. The top one.
It says obtain
Yeah,
level
top
of
one.
relaxed respiration.
69.4.
Oh, that's really...
last
yeah,
one,
that's 3 times normal.
Charge.
Normal's
it on
about
the camera.
22.
That first block.
If you carefully bring it up through the green,
Relaxed respiration.
stop there.
That's the
Yeah.
filtration you're looking for.
55.4.
I consider the first
It's about
sort of
double
2
normal, and then
parts of
carefully
the
bring it up into the
session
clear.
I would practice.
He's really wanting to be in the clear, isn't he?
He's sort of treading this
So once you're
interface.
happy, about 30 seconds.
A little bit higher. That's
normal
it.
respiration. You can try other
22.
things, like describe
That's
what
normal.
happens to your respiration.
That was normal
when you
yesterday.
when you're talking.
So that's quite a convincing
Yeah.
display of behavioural osmoregulation, isn't it?
So
The animal.
normally, if the salinity was the same all the way down, their natural behaviour
And
would
more
go down
observers
to
of
the
you might have
substrate.
noticed a
So
couple of
wave
biscuits
predators.
about from the
But
bench.
clearly he doesn't like this really high salinity.
Now they're in the back for a
It would be
reason.
quite energetically
This
expensive
will
to have to
clean
pump all that
every
into
day.
salt.
This is still at the bottom.
So what he's doing
So
behavioural
make sure that you don't take
osmoregulation.
the biscuits out
So
the
he's
bag.
just swimming to stay in the layer with a
Make sure
salinity
that
which
the
he's sort of
cats
most
within
adapted
the
for, the
bag
salinity
so
that
you
most closely matches his internal
And what
body
you're going to do
fluids.
is
So you could do, if you were a third year, you could do a behavioural study on the amount of time you spend in each layer
chew one of the
and measuring
biscuits and then
salinity,
swallow
and then you
and
can
see
vary
what
pH.
happens.
You could
Okay,
emulate
see how
agricultural runoff by
your
varying
breathing
nitrate
pattern
and phosphate
changes.
concentrations in the water. As an
There's a couple
experiment,
of
a lot of
questions
it's
for you to
actually
look
quite,
at there as well.
you know, you can do quite a lot with it. Look at light dark cycles,
Third part
lunar
experiments,
cycles.
getting into the more
And even just
physiological.
pre-adaptation. So if you kept him in a different salinity and he'd adapted was sort of pre-adapted to the higher salinity, would that
So first of
change
all,
which
you're just
layer
going
he
to
was
stop
in if you
breathing
got regulated all his iron pumps?
Again,
That
see
might
how long
be
it holds you
something
there.
that you could look at.
Put into the comments line.
Do you think he's illustrated the point to us?
Keep track of this later.
Okay.
Do we all believe in behavioural osmoregulation?
It's after a deep
Yeah?
inspiration.
Are we ready to do
again,
some
just
chemistry?
breathe normally, breathe in, hold that breath for as long as you can. Third part is
Yeah, it's still doing it. It's just sort of treading the interface.
Yeah.
But because
I
they've got,
wonder if
they've
you
got
feel that the dye is giving them a little bit of protection, you know.
possibly because they do have very good vision and they will initially go where it's darker to get away from predators.
Force deep breathing, hold your breath.
But this breaks down after a while because he's got these busy little swimmerettes he's propelling himself with that mix all the layers up eventually.
In your lungs.
Yep, he's still
How
at
they
it.
will react to chemoreceptors pick up
Okay.
signals from the body
Do you want to catch me and put it
change
back?
your respiration.
Okay.
So not only in these exercises should you be looking
But
at how
try not to disturb the
That's
layers.
really good.
It's quite tricky.
okay.
Yeah.
After breaks and see how your pattern of breathing compares after the breath hold compared with before. The fourth part of this exercise is to repeat part C, but for the last 15 seconds you're going to breathe pure oxygen and Douglas bag. Douglas bags